Hello everyone, my name is Gary O'Mealy, and in this video we are going to build off of our previous discussion on osmosis and talk about tonicity. So we know that osmosis is going to involve the movement of water from place to place in the body across membranes in situations in which there is an inequality in total solute concentration or osmolarity. So the idea here is that since osmosis will affect the volume of cells of body fluid compartments and therefore the concentrations of their contained solutes, usually in most cases osmosis is not something that we typically want to happen on a frequent basis outside of some very specialized situations such as what's going on in the kidneys most of the time. So the tonicity of a solution or a bodily fluid describes the effect that solution will have on the shape and volume of a cell if that cell were placed into that very same solution. So to introduce this, we're going to operate under the assumption that the osmolarity of the intracellular fluid is 300 milliosmolar, which is the number that we introduced in the previous video. So, if the extracellular fluid osmolarity, which you can see here is labeled in light blue, you can see that a cell with an ICF osmolarity of 300 is dunked in this solution. If the ECF osmolarity is less than 300, in this case 100 milliosmolar, then the cell will swell as it absorbs water. In this case, water moves from low solute concentration to high solute concentration as we established before. So water is going to rush into the cell, and this is going to increase the volume of the intracellular fluid, and that is going to cause some problems. The extracellular fluid in this case, we would say, is hypotonic to the cell. So the problem that I mentioned before is that as the volume of the intracellular fluid gets greater and greater, this is going to cause a buildup of osmotic pressure inside the cell, and the cell membrane, as fascinating and wonderful as it may be, only can contain so much pressure. So eventually we get to the point where the mounting osmotic pressure punctures the cell membrane, and this is going to cause a problem for a cell. So this process is called cell lysis, in which the cell membrane punctures for whatever reason, and this is going to kill the cell because important components of the intracellular fluid are going to leak out into the extracellular fluid. Okay, let's consider the opposite scenario. Let's say the extracellular fluid osmolarity is greater than 300. In this particular case, it's 500 milliosmolar compared to the normal 300 inside the cell. The opposite problem will occur here of water leaving the cell by osmosis because, again, water is always going to move from areas of low osmolarity to areas of high osmolarity. So water will be leaving the intracellular fluid to escape into the extracellular fluid. In this case, the extracellular fluid with the higher osmolarity is a hypertonic solution. So hypertonic environments are also very much undesirable because they cause cells to shrivel up or undergo crenation, and this will lead to cell death as well. So as I said, osmosis is not usually something that we want to happen to most cells under most situations, and for that reason, body fluid osmolarity is typically something that's kept under very tight regulation to make sure that it doesn't get too far off the set point of 300 milliosmolar. So the desirable situation we will want for most cells in the body is what is illustrated here. When the osmolarities of both the extracellular and intracellular fluids match each other, then no net osmosis will occur, and the cell will maintain its volume, its shape, and most importantly, the cell membrane will maintain its integrity, and the cell will be able to continue living and performing its important bodily functions. So this type of fluid in which the extracellular fluid has the same osmolarity as the intracellular fluid, this is an isotonic solution, and this is what we want in most cases for most cells in the body. So I mentioned that osmolarity of bodily fluids, especially the blood plasma, is something that is kept under very tight control through a variety of different homeostasis mechanisms. So we, of course, carefully maintain this through the typical negative feedback that we discussed in the very first video in this human physiology series. So here you can see a rough outline of how homeostasis of osmolarity is maintained. The stimulus that is going to activate this pathway would be when the osmolarity of the blood gets too high, such as what happens when someone becomes dehydrated. This is going to activate sensors in the body, which are located in the hypothalamus of the brain. These sensors are called osmoreceptors, and they more or less are constantly sampling the solute content of the blood plasma to detect when solute concentration gets too high. These osmoreceptors will activate control centers in the hypothalamus and the posterior pituitary. That leads to the secretion of a hormone called antidiuretic hormone, sometimes also called arginine vasopressin. This hormone travels through the bloodstream to the kidneys, which are always functioning in filtering the blood and producing urine. Normally, urine production involves taking excess water from the blood so that we can rid our bodies of that excess water by urination. But in cases of dehydration, that is obviously not what we want. So in this case, antidiuretic hormone acts as a signal instructing the kidney effector cells to stop their filtration or their excessive loss of water through their activities. Water is reabsorbed from the urine back into the bloodstream, and this will help to slow down the dehydration process. This all happens in concert with a separate pathway that causes us to feel the sensation of thirst. So the real way that we bring this back into the normal range is by drinking water, but this certainly keeps the problem from becoming worse than it already is. So as I said, the idea here is that excess water is always being filtered out of the blood by the kidneys, but sometimes the kidneys in this case have to be instructed to preserve water by reabsorbing that water back into the bloodstream. Okay, so with all that being said, let's talk about something a little bit more practical, that being IV fluids. So IV fluids and IV fluid administration is something that every hospital and healthcare worker is going to have to get familiar with and be considering on a daily basis. So in practice, it's going to be important for us to remember our tonicity and osmosis principles anytime you give an intravenous fluid to a patient, given that these fluids are more or less ported directly into the bloodstream. And recall that the blood plasma has a set point osmolarity of 300 milliosmolar, and that is what we want to maintain no matter what we're doing. So, as I said, these fluids are ported directly into the bloodstream, and because we are porting a solution that has its own osmolarity value directly into the bloodstream, that fluid has the potential to change the osmolarity of the blood plasma. And considering that the blood plasma contains the all-important oxygen-transporting red blood cells, if we do anything to change the normally isotonic blood plasma into something that is either hypotonic to make the red blood cells burst or hypertonic to make them shrivel up, that is going to negatively impact oxygen transport throughout the body. So we need to be careful to pick an IV fluid whose osmolarity and solute composition will not cause that to happen. Okay, so here is a short but definitely not exhaustive list of some very common IV fluids that you will find in healthcare settings. If we look at the first one, this is 0.9% sodium chloride, sometimes called normal saline. If you do some quick chemistry math, which you don't need to do, but if you want to, you'll find that 0.9% weight by volume sodium chloride ends up being 150 millimolar sodium and 150 millimolar chloride. Add those together and you get 300 milliosmolar sodium chloride. which is the total osmolarity, which you will notice is equal to the typical 300 we find in most bodily fluids. This makes this fluid isoosmotic and isotonic to red blood cells, which, if your patient is not dehydrated but you want to push fluids, this is definitely an ideal scenario that will not impact their red blood cells in a negative way. Next, we have half normal saline, which has half the usual concentration of sodium chloride, and therefore only an osmolarity of 150 milliosmolar, making it hypoosmotic and hypotonic to red blood cells. This may have you wondering, well, why in the world would we want to give a patient this? Because this is going to reduce the osmolarity of their blood plasma and possibly cause problems for them. Well, you're correct. We probably wouldn't want to give this type of fluid to a patient that has a normal osmolarity of their blood plasma. But if you have a patient who comes in with severe dehydration, this might be a good option for quickly replenishing lost water into their bloodstream if we can't get them to drink water. And then finally, a very interesting one here. This is normal saline, so 0.9% sodium chloride, plus 5% dextrose. This is sometimes called D5 normal saline. This is the same thing as the normal saline we saw before, except it also has 5% dextrose, which is basically the same thing as glucose. So this is what you would want to give a patient if you're wanting to push fluids, but also give them some kind of nutrient to work with, possibly if there's some reason why they can't eat for whatever reason, because we've said before, the brain and the brain cells can only feed on glucose, so we need to make sure that we're keeping this patient's blood glucose up. The issue, though, is that this solution is 300 millimolar sodium chloride and 300 millimolar dextrose, giving it a total osmolarity of 600 milliosmolar, which makes it hyperosmotic to the blood plasma. However, the thing that is very interesting here is that this is still an isotonic solution. And if that's confusing, you have to remember that tonicity refers to the effect that a solution will have on cells that are exposed to it. So even though this solution has a very high osmolarity, it's not going to cause cells or red blood cells to burst. Here's why. That glucose is not going to stay in the blood plasma for very long. The purpose of glucose traveling through the blood is to get absorbed by cells throughout the body so that they can use that glucose for cellular respiration. So, after that glucose gets absorbed by the body cells, that glucose will no longer be in the blood plasma, and all that's going to be left behind is the 0.9% sodium chloride, making, in effect, the practical and effective osmolarity of this solution 300, because that 300 worth of glucose is not going to stick around in the bloodstream for very long. It's getting absorbed by the body's cells. So with all this in mind, choosing an IV fluid has to be done with very careful consideration of a patient's current condition. So something to think about in the future. All right, that's going to wrap it up for this video. Here is a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, how does changing the osmolarity of the blood plasma affect red blood cells? Next, if pure water were poured into the blood intravenously, how would this affect oxygen transport? And finally, physiologically, why does dextrose not affect the tonicity of IV fluids? All right, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you next time. So long.